You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you've enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you. Hello, this is Richard Jacobs with the Future Tech and Future Tech Health podcast. I have Dr. Young-Hae Sung, a postdoctoral associate, University of Florida, biomedical engineering in the Schmidt lab. So, Dr. Young, thanks for coming. How are you doing?
Good. How are you?
Good. Tell me what kinds of things happen inside the evil Schmidt lab. What goes on there?
So our lab is focused on utilizing naturally derived biomaterials to develop in vitro disease models, as well as creating scaffolds that can help nerve repair. So natural biomaterials, we largely use hyaluronic acid-based hydrogels, as well as recent focus has been on using decellularized tissues. So that's what we do.
Okay. I had thought that nerves don't really regenerate or repair themselves. Do they? Is it just that they lack a scaffolding, or what are the missing components to allow for nerve regeneration in the body?
So it depends on the system. For central nervous system, the nerves don't regenerate spontaneously. And in case of injury, that's particularly because of the extracellular matrix proteins that are deposited after the injury, specifically inhibit axonal regeneration. So therapies that have worked in various literature include enzymes or proteases that specifically cleave these inhibitory extracellular matrix proteins. So that's largely for the central nervous system. For peripheral nervous system, up until a certain injury length, the neurons can regenerate by themselves.
Why do you think the central nervous system won't regenerate? Why is this extracellular matrix created? Is that the body's way just of trying to heal and isolate the damage, but it doesn't account for the need for nerve regrowth?
So glial cells, such as astrocytes that are present in brain and spinal cord, upon injury, they become reactive, and then they actually deposit glial scar filled with chondroitin sulfate proteoglycans or CSPGs, which are the inhibitory components, but also the myelin sheath in the central nervous system, which is created by oligodendrocytes. It breaks down, and also the breakdown of those myelins are inhibitory as well. Whereas in the peripheral nervous system, the Schwann cells, which also create myelin sheaths, these cells produce different types of ECM proteins, and the ECM proteins that are derived by Schwann cells actually promote nerve regeneration. So I think it's a combination of different cellular compositions in central versus peripheral nervous system that's eliciting different responses.
So I guess there's cell-to-cell communication between the existing nerve cells and this newly created matrix, and the matrix is telling the cells don't grow?
Yeah, that's what's happening in the central nervous system, like for spinal cord injury, for example.
Oh, okay. So what's your goal? Is to replace or inhibit the current extracellular matrix so that the signaling doesn't occur, or the growth of the matrix itself doesn't occur? What's the goal?
So a lot of the initial work on spinal cord injury repair done by the Schmidt lab has focused on delivering, utilizing the biomaterials such as the hydrogels derived from peripheral nerve hydrogels, because as I said earlier, peripheral nervous system they can regenerate, the nerves can regenerate because of the differences in ECM components. So what the Schmidt lab has done is you take out the nerve, the peripheral nerve, and then you decellularize them, meaning you subject the nerve to a series of different treatments to remove the cellular components while retaining the extracellular matrix proteins. And then after that, you can digest these decellularized nerves to create injectable hydrogels. And you can take these pre-gel formulation and then inject it into the spinal cord lesion, because what happens after spinal cord injury is that you get this lesion cavity that's basically just a hole in the middle of the spinal cord. So you can fill it with these injectable hydrogels created from the decellularized peripheral nerves. Does that make sense?
I'm not sure if you can maybe just explain it once more, like in a little slightly different way. Yeah.
Okay, so like earlier I said the peripheral nervous system, the nerves can regenerate spontaneously, and because that's because of the different ECM components in the peripheral nervous system versus central nervous system. So what our lab has recently done is, so our lab, let's take a step back. Our lab is known for developing a technique to decellularize peripheral nerves. So what the Schmidt lab has done earlier, like over a decade ago, is you take sciatic nerve from a rat, and then you treat it with detergents and other buffers to remove the cells while retaining all the extracellular matrix proteins. So what we have started doing recently is that you take those decellularized nerves and then you digest it in acid solution containing enzymes or proteases. Then you get this, then the nerves become like homogeneous solution, like the nerve and the acid enzyme cocktail becomes this homogeneous pre-gel solution. And then this becomes hydrogels after elevating the temperature as well as the pH of the pre-gel solution. And this pre-gel solution, like before you elevate the temperature, you can neutralize the pH of the pre-gel solution to make it ready for gelation, like basically polymerizing to become this like jello-like structure. Instead of like water turning into jello, something like that. So what you can do is you'll take this water-like structure with neutralized pH, and then you take it into a syringe, and then you inject it into a spinal cord lesion, which is actually a cavity or a hole in the middle of the spinal cord. And since it's in a body temperature, this pre-gel solution from decellularized peripheral nerves, when the temperature of this gets elevated to a body temperature, it'll turn into a gel, and then it'll start to fill the spinal cord lesion cavity. And the goal is that we can use this strategy to entice the severed neurons in the spinal cord to regenerate across the lesion sites, but also we can engineer these hydrogels to contain cells or other therapeutic molecules to deliver additional therapeutic benefits.
So you're seeding a man-made ECM with the components necessary for the central nervous system to act like the peripheral nervous system and regrow and innervate a damaged area.
Yeah, in a way, but we're not developing the material from scratch ourselves. So we would take the peripheral nerves. A lot of our work has been done with rat sciatic nerves. So we would take the nerves from a rat, and then we process that to develop our injectable hydrogels.
Does this migrate very much? Like when you inject a damaged site, what's the radius at which it has an effectiveness? Do you need to inject dozens or hundreds of times in a small radius for this to work? And does it migrate outside of the area? And, how well does this work so far?
Oh, no, the gel doesn't really migrate outside of the cavity. It just fills the cavity and it stays in there. And if anything, it'll be degraded over time. And then for the rat spinal cord injury model, which we have tested our hydrogels, we only need to inject like 6 microliters of this pre-gel solution. So it's a very small volume that we're dealing with here.
So how would you do this in people? Would you have to sacrifice part of a nerve, like one of their peripheral nerves that they didn't care about, and then create these gels and seed them and then, inject the spot of real trouble? Or how would this happen in people?
No, ideally you want to take a donor nerve. That way you don't create like additional donor site morbidity in the patient.
Okay.
So earlier I said Dr. Schmidt has developed this deserialized technique for peripheral nerves, and her technique was actually licensed by a biotech company called Axogen, which is actually based near Gainesville, Florida. So we have, there are ways to obtain the nerves from Axogen and then work with those nerves to create these hydrogels. So I imagine that's how it should be done for human patients. But we haven't tested. I don't think we're anywhere near testing this platform in humans yet.
Well, why not? If why wouldn't there be an expedited path, through the FDA for people that are paralyzed, because of a spinal cord injury?
Mm-hmm.
It seems like what harm would come to them by trying this? Even if you did if you cannibalized, let's say they had like a T1 problem and they were quadriplegic.
Mm-hmm.
What harm would come from trying to harvest something from like their toe or their ankle or some a nerve there, and then try to seed the area that's damaged to try to restore most of their function?
Oh, that's a good question. I mean, like with the spinal cord, like patients suffering from spinal cord injury, their immune response is generally weaker. So I'd imagine trying to perform additional surgery just to take those nerves out of their body. I think that itself would be a pretty challenging case.
Yeah.
And then we also don't know if they're I think we would need to make sure the nerves taken from the spinal cord injury patients retain similar ECM proteins, and they can be processed similarly.
Have you identified in the rats so far what specific proteins or chemicals are causing the regeneration? Have you isolated those to see what they are?
We haven't tested those specifically, but generally what the field I think largely agrees upon is collagen and laminin are the main ECM components that promote neural regeneration.
Oh, have they tried to just isolate those two and put that into a gel without taking it from the outside nerve?
Like not specifically from the nerves themselves, but I mean there are ways to easily make hydrogels that are composed of collagen and laminin. But I am not aware if people have done that specifically for spinal cord injury repair.
Yeah, I would think that would be an easier, or a good confirmatory path, so you don't have to harvest that sciatic nerve.
Yeah.
Then you don't need to harvest anything. You could just culture, you can culture other cells and produce those two substances.
Yeah. So a lot of people do like culture cells like fibroblasts that deposit a lot of ECM and then harvest those cell deposited ECM to make scaffolds. So that's definitely a possibility. So but I don't think people have done that specifically for spinal cord injury. I think people have used it for other tissue regeneration applications. This whole biomaterials, hydrogel-based approach to repair injured spinal cords, I think the whole concept is pretty new in and of itself.
So what happens right now in a central nervous system lesion? Is it just that extracellular matrix forms, but it has no growth factors? It has inhibitory factors in it, but there's still scaffolding there? Or is it random and it's not? Is there no apparent scaffolding there, even if there were growth factors?
Like in regular spinal cord injury cases?
Yes. Yeah.
So over time, you get this lesion cavity formed, and it's surrounded by a glial scar, which is filled with those inhibitory ECM components. And then the lesion cavity is filled with fluids. And also the glial scar barrier in and of itself inhibits neurons from crossing the cavity to regenerate across. So I mean, like there may be growth factors, but the presence of the glial scar in and of itself is preventing the neurons from traversing across the lesion site, so to speak.
Okay.
Yeah. So so there was like close to a couple of decades ago, there was a breakout paper where a bacterial-derived enzyme called chondroitinase ABC was shown to specifically cleave this these CSPGs and allow the neurons to regenerate across the lesion. So there's been a lot of research on using biomaterials to better deliver this enzyme to help remove this barrier, to break down this barrier.
Okay, I was just wondering if the nature of the ECM in the spinal cord when there's a lesion is the nature of the ECM itself. Could it be a scaffolding, or does it have to be reconfigured in such a way that it would allow it would become scaffolding? I know it's devoid of growth factors. It actually has inhibitory factors in it, but again, is it still considered a scaffolding?
I don't think so. I don't think you would call that scaffolding, no.
Okay, I was just wondering if it somehow had that characteristic, but I guess not. So
no. I mean, at least my understanding is that like for scaffolding, you would have to like inject it or implant it somehow, or at least that's my
Right, structurally it would have to change somehow. I got you. Okay. So what stage are you at with the rat experiments? Like, are you at a point where it's working and you want to move to the next stage, or how much work is left, do you think, to make it a viable process, at least in rats?
So our lab has recently published a couple of papers showing the feasibility of this hydrogel platform. First as like on its own, the hydrogel on its own, and second as a delivery carrier for Schwann cells. So our lab is actually now testing a variety of different strategies to utilize this nerve-derived hydrogels to deliver growth factors or other therapeutic molecules or other cell types. And I'd say those projects that are currently ongoing are pretty new at this stage.
Okay, so they're pretty new. So you think it'll be a number of years before you're able to go to the next level?
I think so, yeah. I think so, yeah. Okay. We're just starting. And also,
well, I was going to say, what do you think are some of the remaining challenges that are important?
Let's see. I think, so a major issue with biomaterials-based approach is that you can entice the neurons to regenerate into the scaffold, but it's harder to make the axons grow back into the native tissue because they like the scaffold so much. That's one of the major issues with biomaterials-based approach for spinal cord injury repair, and I think there needs to be a way to better design the scaffold in a way that the neurons will not only want to start regenerating into the scaffold, but also extend out back into the native spinal cord tissue to form correct synapses and actually promote full or near complete functional recovery.
So it sounds like not the scaffold is important, but also at the edges of the scaffolding, the transition to the currently healthy tissue is it's super important to have the right transition so that it actually does it. So like I think a lot of the axons that do regenerate into the scaffold tend to just stay in there. Yeah, so we need to find ways to make them go beyond. Okay, gotcha. Well, very good. So what's the best way for people to find out more about, what's going on in the Schmidt lab and, ask questions and things like that?
So Schmidt lab, we have a lab website. So if you just Google Schmidt lab, University of Florida, you'll find the link. So there's one way of staying up to date on what's going on in our lab. And also our PI, Professor Schmidt, also runs a Twitter account for the lab. So that's another way of finding more information on our lab, current events in our lab.
Okay. Well, very good. Well, Dr. Young, I appreciate you coming, and I really hope that this technology, does well pretty quickly because it's super important. A lot of people need the help, so I appreciate you coming.
I hope so too. Thank you.
You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you.